Thank you very much, Lionel, and obviously I'm really grateful that you consider me as a speaker today, and it's also very nice to see a lot of familiar face and good friends today. So I will try to give you some sort of input in this alternative model I will present today, which is regarding an alternative view of cells, right? That will be my very first part of the talk. Then I will just address the model system we use to test some of the predictions of this new model. And I will just show you some sort of modeling with it and some connections of this model with the cellular membranes. And finally, I will just try to address the conclusions. Um, well, the bottom line or the first things I will like to pose to start the talk is that the canonical model, the dominant model of the cell, is basically derived from schemes that are completely based on the physical chemistry of dilute systems, right? These are isotropic environments. And unfortunately, this is the only vision that is taught in most of the courses in cell biology and biophysics, right? This is involved mass action kinetics, dilution solution theory, for example, Van Hoff, where intracellular water is seen as a biological solvent. Also, you consider that inside the cell, there is a complete ionic dissociation, right? Diffusion models based on Brownian motions are the ones used, and this also implies that the random thermal collisions will dominate the motion inside the cell, right? This lead to the steady state energetics, so you will need high energy consumption in order to keep the system in a steady state, and of course dissipation of energy is the process that dominates all these scenarios. In this model, there is a central role of membranes, right? And the concept of permeability through semi-permeable membranes, right? And of course, most of the molecular events are basically going through the single molecule in order to explain phenomena, right? So it's not that you really take into account emergent properties in the system. This leads essentially to this picture I put you here, where you have free water, free ions, and there is a membrane that divide compartments. This is what is taught. But of course, there is an alternative view, and I will just try to put forward to you. One point that is very important in all these concepts regarding the canonical model, although it is acknowledged the molecular crowding and the spatial confinement that happens in the cell, right? And actually, that is not really incorporated in the model because obviously, in an environment like in the intracellular, right, you have a really high concentration of macromolecules with lead a concentration of macromolecules around 200 to 350 milligrams per milliliter, which is a lot. And that is, of course, will impact somehow in the physical state of intracellular water, which is by the way, the most abundant component in the cell, right? This will lead, obviously, if one analyzes from basic physical chemistry, a very big impact in the colligative properties of the system, and of course, in the properties of intracellular water, right? And well, also it leads to some thermodynamic consideration if you really can consider the system as purely dissipative. When I'm talking about crowding, I'm talking about something like I show you here. You can see this is a sketch of the interior of a mycoplasm. And you can see that if you really try to locate water molecules, in between the macromolecules that you have inside the cell, in one dimension, you are talking of no more than 10 molecules of water, right? This is just to illustrate, this is also a very beautiful experiments that Matthias Clausen did in Denmark, where he labeled also actin in the cell. And actin is one of the many proteins that exist in the cell. So you can get an idea about the crowding effect here, right? Well, all these sort of you know, considerations lead to a question, right? And the question is, to what extent you can really assume that the behavior of macromolecules, ions, water in the intracellular environment can be described by ideal solution theory, right? Basically, what is happening is that you know, you are not leading a room to another view, because generally what happens if you have deviations of this solution theory, right? You will try to add correction factors, like, for example, diffusion to anomalous diffusions, without changing the conceptual frame. And the other question I will try to put forward in this talk is if it is correct to assume that intracellular water is a passive element of the cell and acts only as a solvent, right? As it is teach or as it's taught in the courses. Well, there is a protoplasmic doctrine, which is an alternative view of the cell. Actually, they are kind of mutually exclusive if you compare with the membrane theory, which is the canonical model, right? And this is contemporary, actually, with the membrane theory. If you go to the history, at the moment that they were posing that osmotic effects are happening in cell in the 19th century, there were two views. One, there was a semi-permeable membrane, and another one that all this process is lead to the properties of the cytoplasm. Here, there is a very strong principles of colloidal physical chemistry and statistical mechanics. This is really formal and quantitative theory. Basically, it will assign a central role to the colloidal nature of the cell interior. So membrane is not at the center of the model, right? It's also used the concept of colloidal water or polarized water, which is led by the experiments of Graham in dialysis way back in time. And of course, it's put a lot of emphasis in the emergent properties of the systems, right? Instead, to talk about permeability, in this theory, you are talking about partition, right? And essentially what it is telling is that these solutes, ions, and other you know, substance in the cell will accommodate by adsorption to fixed sites inside the intracellular environment, which are essentially polyelectrolytes, can be intracellular proteins, can be DNA, can be RNA, and all these other things, right? It certainly take into account the crowding and the confined nature of the environment, right? And they consider transitions between two states of low entropy, right? And here, different to the other model, there is insensitivity to random thermal collisions. So you have a coherent sort of system. The most complete version of this theory appears in 1962, and is known as the association and induction hypothesis. Of course, I cannot go to all the details on that because it's not time to, but I will just try to point it out which are the most relevant points for this theory. Well, just to make an analogy, you know, the protoplasmic doctrine will see the cell essentially as a gel type of different to membranes containing aqueous environments. Well, which are the general features of this theory? Well, first of all, I will put forward that this a quantitative theoretical framework based on the concept of fixed polyelectrolytes in the cells and association process with ions, right? Where crowding and confinements are really important issues. There is a really large a large body of experimental evidence done by several groups in the 60s and the 70s, and it essentially established mechanisms that explain coupling observed in cellular systems, for example, between physical properties, chemical properties, physiological aspects. And of course, it's emphasizing the emergent properties, right? Instead to see the cell as a you know, steady state, stationary steady state, you know, dissipative process will define the living state as a metastable equilibrium between two states. One is in the resting, another is in the actives. And both of these states are in a low entropy states. And they, as another important thing, requires very small energy inputs to maintain the dynamical properties. This theory applies very well to explain all this phenomenon of liquid immiscibility that is very, very well discussed these days, right? Basically, you have, they consider the major component of the cell, water, proteins, ions, monovalent ions, particular potassium, as an amalgamate system. So they are electronically associated and they will interact directly or indirectly with other cellular components in a sort of coherent integrated system. It's like a, you know, sort of amalgamate responsive element, right? This is how we see in the cytosol. And the very interesting thing concerning this is that the global physicochemical state or the physical state of all this amalgamate system will be regulated by metabolism through association phenomenon and inductive electronic effects. This is a very large and very concise theory on these effects. And the key intervenings concerning inductive effects are ATP, hormones, and some divalent cations of calcium, and the interactions with the intercellular proteins. And this is leading to another important thing, which is that water, instead seen as a passive component, will be a reversibly dynamical structure active participant, which the rotational dynamics of water will be regulated by these associations and electronic inductive effects with, of the metabolites with the proteins. According to this theory, depending on the metabolic state, you will have more or less polarized water. Essentially, that implies a change in the rotational dynamics of water and as a consequence of the chemical potential, which will be a very important point I will address at the end of the talk. Well, the theory is, will quantitatively explain the four paradigms of the cell physiology, for example, cell volume control, selective ion distribution, selective solid partition, and of course, cellular electrical potential. And the most important thing that the theory still remains unrefuted. Well, what is our experimental system and why we choose this experimental system? Because the whole idea here was just to test some of the predictions of this theory. And what we do, or what we use, are suspensions of yeast, Saccharomyces cerevisiae, which show oscillatory behavior in a metabolic process. In this particular case, is glycolysis. This is a very well-known phenomenon. It's more than 70 years has been described and studied quite a lot, right? Essentially, what you do, you synchronize starved populations of yeast by adding, by starvation, and then you add glucose and then cyanide just to kill the respiratory change. And then you follow, for example, NADH and ATP over the time. What you see is that there are oscillations in these metabolites, right? And this is what you measure. Here we use the fluorescence of NADH that is transformed, it is oxidized, and of course the oxidized form is non-fluorescent, so you can see the oscillations of the NADH. Also using sensors, you can just follow oscillations in these metabolites in the ATP. This is an autocatalytic reaction, glycolysis, right? So you generate 2 ATP per cycle, and then of course you will have every time that the cycle is going one way, you will see an oscillation on the activity of these compounds. The properties of these oscillations are from single cells, but when you have a suspension of cells, it became macroscopic because you have a coupling with some sort of diffusive metabolites as acetaldehyde, for example. The very interesting thing about this system is that even though, you know, they are tackling this with dilute theory and talking only about enzymes, is that it has been reported that not only the intervening metabolites, NADH, for example, or ATP, will oscillate, but there is a very large number of cellular properties that will oscillate coupled with the chemical or biochemical reaction. And this is include, for example, pH, intracellular heat fluxes, electrical potentials, and things like that, as I'm showing here. This indicates is essentially that you have a very strong bidirectional influence from glycolysis, right, to the physical properties and vice versa, right? So you cannot ignore that this metabolic process is, some sort affecting the physical and physical-chemical properties of the whole cell. And obviously, as I said, these sort of effects are not basically described by the models based on mass action kinetics on Van Hoff's theory. Essentially, they are completely ignored, right? And so we think that the study of this coupling between these cell properties and the metabolic changes will be a very, very, very important thing to take into account in order to understand how essentially the cell works as an emerging, system. Well, we use this experimental system to, as I said before, to test the predictive power of this association and induction hypothesis. And of course, we did really a lot of experiments, and I will just simply focus in the first one, which is if there is some sort of connection between the dynamics of intracellular water and these biochemical oscillations with ATP. Also, we test effects of the cytoskeleton on the oscillations, the effect of potassium, I mean, if it is absorbed or not, and all the coherence from this thermodynamic perspective, but certainly I will not have time to present all these sort of things. But if you are interested, we just recently published a review article where we discuss all these sort of things together. But as I said, I will just concentrate in the first point. And for doing that, I have been working really for a long time with the dimethylamino-naphthalene probes. These are probes that has been synthesized by Gregorio Weber in the late 70s, and Enrico Graton and Tiziana Parasasi, they incorporate this probe to study the dynamics or phase states in membranes, right? Basically, what they are using is a solvent relaxation model, which has a two-state model, and is giving you some sort of quantitative information about the solvent relaxation process in the membranes, particularly with Lordan, right? And this is essentially defining this generalized polarization function, which is giving you some sort of information between interconversion of relaxed and unrelaxed states, right? So you have a high GP when the system is unrelaxed, and when you have a low GP when the system is relaxed. So we decided just to use this concept of the GP and the applications of these dimethylamino-naphthalene probes, but in the cell cytosol and the membranes, of course, right, of the cell. Essentially, we translate all this methodology to try to see what is going on in the cytosol of a cell when you have these biochemical reactions going on, oscillatory biochemical reactions, okay? Well, basically, we use, as I said, 3 of these dimethylamino-naphthalene molecules. And the reason we did that is because we tried to map environments of different hydrophobicity. For example, Lordan is a highly hydrophobic probe, which is going to go to membranes, while Agdan, for instance, will be more located in the cytosol of the cell, and Prodan has an intermediate behavior. Well, we induce the oscillations in these cellular systems, right? And what we see when we put this probe is that the intensity of these probes oscillate, and not only oscillate, but also oscillate with the same frequency of oscillations in ATP and an ADH. So from this experiment, we can just basically conclude that the temporal behavior of the fluorescence of these probes from Lordan to Agdan, so it seems to be that there is, all the cellular environment responding to this is coupled with the biochemical reaction and shows essentially the same period, okay? And also the other interesting thing that we observe is that if you compare the oscillations in Agdan, for example, and ATP, you will see that these oscillations are completely in phase, which is something important I will discuss later on. The interesting thing is when you add inhibitors of the glycolysis, these oscillations disappear completely. The other experiments we did, because in here we are just measuring, intensity oscillations, is to try to map what happened with the GP function. And the GP function is essentially a model that will, tell us about an interconversion between this relaxed and unrelaxed state. I think that Lionel was talking about this probe, so I will not just go to many details. So this is the experiments we did. This is the NADH fluorescence, this is the ACTAN fluorescence, and this is the ACTAN GP, right? And what we see here is that when we do the power spectrum, the frequency of the oscillations for these two molecules are essentially the same. We confirmed that in experiments in a fluorometer, but also we observed these things in the microscope. We were able to calculate the oscillations of the GP as well. So it's essentially, the fact that you do have an oscillation in the GP function basically is telling you that the solvent relaxation, it is the mechanism behind that. And a very important control we perform with these things is to add deuterium oxide experiments. I did that, I remember when I was working in Enrico's lab as a postdoc, we were just discussing about the solvent relaxation mechanisms in membranes. So in a membrane, for example, you have a phase transition from gel to fluid phases, and then you use the Lordan probe just to calculate the, or to follow the phase transition of the membrane. And now if you put the deuterium oxide in the membrane, right, and you change the mass of the solvent, in this case, in the artificial membrane system, then we observe a reduction in the delta GP in the transition, which means essentially that when you increase the mass of the solvent relaxation will ralentize. So we use this concept just to do it in the cells, and you can see up here, this is the NADH oscillations, the metabolic oscillation. As soon as you start to add deuterium oxide, then you can see that the period of the oscillations are increasing. So the dynamics start to get slower. And this is something that you see not only with the metabolites, but also with Prodan, with Agdan, and other probes. So this is give you some sort of confidence, essentially, right? That the water dipolar relaxation is the parameter affecting this probe. So you can see that if you put this probe in water or deuterium oxide, the spectra is the same. So this is nothing to do with the fluorescence of the probe. It's more to do with the dynamics of the system. And last but not least, we observe both for the metabolites and the DAN probes that these oscillations are scale invariant. When you talk about a scale invariant phenomenon, you're talking about an intensive properties of the living system. So this leads to these conclusions that we achieve some sort of temporal and spatial evidence of the cytosol acting as a medium, exhibiting a very rapid and coherent coupling between this intracellular water dipolar relaxation, which is an intensive properties of the system, and the metabolic process, right? This essentially is disputing the idea that the intracellular water is acting as a passive component, because nobody's taking water into account. We proceed further with these stories, and actually we were very interested to see, or to try to explain how come that also Lordan will oscillate, particularly because this association induction hypothesis theory is not placing any role for the lipids. So I was very curious to see if I can just put the lipids into the scene. Well, we did some mathematical modeling of this. We basically combine what is called the Yang-Link adsorption isotherm, which is general and entirely based on the association and induction hypothesis, and some sort of kinetics models, right, to reproduce, try to reproduce these oscillations between the polarization of water or dipolar rotational relaxation of water we observe in these experiments with the glycolytic oscillation, and we succeed to do that, right? So you can reproduce very well considering this, regulation effect of the ATP on the intracellular properties of proteins. For example, particularly intrinsically disordered proteins and cytoskeletal proteins, okay? And the interesting thing in this is that this model will predict that if you do have an spatiotemporal coupling, right, in the polarized state of the water, from the place where the glycolysis happens, which is, you know, in the cytosol, to regions where the glycolysis is not occurring, which is the case, for example, of the membranes, then you will expect that the waves, the oscillatory waves, will have a phase shift. And you can do the phase plot. And if, of course, you have an in-phase waves, you will have this linear response. And if it's out of phase, you will have this sort of response in this phase plot. Well, we did that experimentally, and this is a very interesting thing because, you know, during the oscillations in glycolysis, it's not only Lordan, which is oscillating, but also Nile red. And this is, of course, Agdan, which is the more hydrophilic probe, right? So bottom line, all these probes will oscillate, you know, indicating with the same frequency, indicating that, you know, the coupling is cell-wide, as I showed you before, which is essentially that will occur in the cytosolic environment, but also in the lipid structures, right? Now, the interesting thing is that the oscillation from these hydrophobic probes are really not strictly in phase with those produced by the hydrophilic probes. And this is what we did experimentally, and you can see here, right? If you correlate the fluctuations of Nile red fluorescence with the damp probes, the different ones, you can see that Lordan is the gray one, the red one is Prodan, and the blue one is Agdan. Basically, this is confirming somehow the prediction of our model, right? So therefore, we need to do an interpretation, obviously, on this coupling. And what we are thinking is that the dynamics of the cytosolic water, you know, which is regulated by these changes in the activities of the ATP, meaning more polarized to less polarized, will be transferred somehow to the interface where you have the membranes, basically the water that is associated with the polar head groups of the lipids, right? So essentially what this is telling you essentially is that these effects on the dynamics of water that you have in the cytosolic environment can be transferring somehow to the membranes. The interesting thing is that when you have this polarization of water, you will change the water chemical potential. So what we are thinking is that you may have some sort of lyotropic effects in the lipid structures mediated by the metabolic changes. And this is the model we come out right now. And of course, it's a working hypothesis. We are doing a lot of experiments right now just to corroborate this. It's certainly something that you can see in the cell. So we are proposing that the structure of these biological membranes will be responding now to metabolic changes via the polarization of the intercellular water. Essentially, this is the whole scheme. I will not go in detail. I have no more time now. So just to conclude, well, the association induction hypothesis will emerge as a very satisfactory theory to explain somehow this mechanochemical coupling that we are observing during this metabolic cell function, and is obviously supporting the view that the cell interior is a colloidal system and not just a dilute solution, right? In here, the intracellular water will be capable somehow to act as a reversibly dynamically structured participant, establishing some sort of coherent fast response on length scales that comprise essentially the whole cell, are much more faster than, for example, diffusion, right? Finally, we somehow incorporate lipid membranes to this hypothesis, right? And we propose essentially that membranes can be responsive structures to metabolic changes that you have in the cell. And finally, as I mentioned at the very beginning of the talk, this theory is certainly suitable. It's giving you a very interesting theoretical frame from the physical chemistry of colloids to describe the emergence of these molecular condensates, where obviously polyelectrolytes interacting with water, interacting with ions, it really take a very big role, but nobody is really focusing in that. So, well, thank you very much for your attention.